Hi, my name is Eva Solis. I'm part of Dr. Trent's summer course for biology, and I'm going to be going over DNA replication. I'm going to start off with the definition of DNA replication. It is the biological process of producing two identical replicas of DNA from one original DNA molecule. Facts about DNA replication include that it is semi-conservative. They are complementary, so A goes to T and G goes to C, and they are anti-parallel. So it'll go from 5 to 3 and then 3 to 5. There are two different methods of replicating DNA for prokaryotes and eukaryotes, and I'll be going over that towards the end. And there are three main steps, so it's initiation, elongation, and termination. The keywords are helicase, replication fork, leading strand, lagging strand, DNA polymerase, primase, RNA primer, and Okazaki fragments, DNA ligase. So the first part of DNA replication process is when the helicase unwinds the double helix. Helixes lose hydrogen bonds, creating the replication fork. So this is the helicase creating the replication fork. This divides the leading strand and the lagging strand. Then DNA polymerase adds matching nucleotides to leading strands. The DNA polymerase also scans for mistakes in matching. But before polymerase can add nucleotides, RNA primer must be placed in the beginning of the hook of polymerase to hook the polymerase. So right here, to hook it to go on with the nucleotides. And since DNA polymerase can only read 5 to 3 in the leading strand, it has trouble with the lagging strand, which is 3 to 5. The help of primase creates sections that are called the Okazaki fragments, so it can work backwards. And finally, enzyme DNA ligase goes over Okazaki fragments, sealing nicks and converting it to DNA strands. The difference between prokaryotic and eukaryotic replication is that prokaryotic replication occurs in opposite direction at the same time. There is no nucleus, so it happens in the cell cytoplasm, and prokaryotic cells have one to two types of polymerases. They also have circular chromosomes, so there is no end to the synthesis, and it is continuous, while eukaryotic replication has multiple points of origin and unidirectional replication. The process occurs in the nucleus of the cell. Eukaryotes have 4+ polymerases, and chromosomes contain telomeres in their ends and replicate only in S phase of cell cycle. And that's it.